Good morning, everybody. So I'm going to start with first session today, and our first speaker is Jordi Clarimon. So he's an investigator of the genetics of neurodegenerative diseases unit at the Research Institute of Sant Pau Hospital in Barcelona. She is a Cybernet research member and an investigator. His research is focused on the discovery of genetic factors contributing to neurodegenerative diseases, dementias, ALS, and Parkinson's disease especially. He's as well coordinator of the Dementia Genetic Spanish Consortium. So please, Jordi, you are welcome. His talk is entitled Using Genetic for Biomarkers Discovery and to Unmask the Genetic Architecture of Neurodegenerative Disorders. So please, Jordi, thank you for coming.
Good morning, everyone, and thank you, Teresa, for this presentation. And I would like to thank the scientific committee for allowing me to present our data in this auditorium. So I'm going to divide this talk into main sections. During the first section, I'm going to talk about how we can use genetics to understand the biological basis and the biological boundaries between neurodegenerative diseases, and how can genetics shape the clinical course of these disorders. And during the second section of this talk, I'm going to talk briefly about how can we use molecular genetics to design novel biomarkers for neurodegenerative disorders. Well, I've been told. Well, yes, sorry. So, as you all know, there's a considerable clinical and pathological heterogeneity in neurodegenerative disorders, and we have used this huge heterogeneity to classify these diseases. And in this sense, genetics is helping us a lot to define these boundaries. Genetics is helping to define both the similarities and dissimilarities between diseases. And I'm going to take a couple of examples about this. So the first example is on, I don't know, this is, should I point toward? No sé, això no acaba. Well, so the first example, I'm going to talk about Lewy body dementia. Yeah, I'm pushing this. About Lewy body dementia and Parkinson's disease. I mean, I promise I'm pushing the next button. I mean, it's not. I'm sorry about that, but is there any other? See one. So these are the most recent genome-wide association analysis on Lewy body dementia performed by Rita Guerreiro at the UCL and by Mike Nails at the NIH for Parkinson's disease. And as you see here, there are quite a lot of differences, but also important similarities between both GWAS. And for example, well, this hit here in chromosome 4 stands for alpha-synuclein, and this other hit in chromosome 1 stands for GBA gene, and is well known. As demonstrated by, for example, this multicenter analysis performed by Ellen Sidransky in 2009, that GBA is the largest genetic risk factor for Parkinson's disease. And what she found through the analysis of this gene in more than 1,000 patients with non-Ashkenazi origin, she found that 7% of these patients harbor heterozygous mutations in the gene. And interestingly, I mean, maybe it's me, but so what she also found is that mutation carriers may be more prone to cognitive deterioration during Parkinson's disease course. So what we aimed to do was to replicate these findings in a Spanish cohort of Parkinson's disease patients, but not only this, but also to infer whether GBA mutations could affect the clinical course of disease, and also to test the GBA in Lewy body dementia. So this is a work performed some years ago by Nuria Setou, a PhD student in our lab, and she fully sequenced the coding region of the GBA in a comprehensive series of Parkinson's disease patients, 225 patients that from the movement disorders unit in our hospital, and that were followed up for an average of 12 years. She also analyzed a cohort of 17 patients with Lewy body dementia, and in this case, they were pathological diagnosed patients, and she also included healthy controls, as you see here. So this is what she found. She found that 10% of Parkinson's disease patients in Spain have a mutation in GBA, and these two mutations, the L444P and the N370S, are the most prevalent allelic mutations in our population. And she also found similar percentage, actually 12% of patients with Lewy body dementia have a mutation in GBA, whereas only one single control, representing less than 1%, also presented this mutation in this gene. So interestingly, when we analyzed whether the GBA mutation patients have different clinical course, we found that patients with a mutation in GBA have 6 times the risk to develop dementia compared to patients without a mutation. And this is telling us that assessing the GBA gene in patients with Parkinson's disease could have profound effects in clinical practice. So following this idea of using genetics to better shape the biological boundaries between diseases, I'm going to focus now on 2 other diseases. These are amyotrophic lateral sclerosis and frontotemporal dementia. And as you know, these 2 diseases are can be conceived as the end of a phenotype continuum, of a disease continuum, and in which only 50% of patients with ALS have only motor neuron disease, whereas the other half of patients with ALS have some kind of cognitive deterioration early in the disease course, and 5 to 10% of ALS patients also fulfill the diagnostic criteria for frontotemporal dementia. And the same is true for frontotemporal dementia, which a third of patients with primarily diagnosed from frontotemporal dementia also develop some kind of motor neuron disease, even if this is subclinical. And this is the genetic landscape of this continuum. And as you see here, there are some genes that are related mainly in ALS. There are other genes that, of course, are related to frontotemporal dementia, but there's a substantial number of genes that can be related to both diseases. And above all genes, C9orf, as you may know, is the main genetic cause for both ALS and frontotemporal dementia. Actually, it represents this expansion of the expansion of this exonucleotide that is located in the non-transcribed section portion, 5' portion of the gene. So subjects with an expansion of above 30 repeats are very at risk of developing either ALS and frontotemporal dementia. And interestingly, there's a north to south gradient of the prevalence of this alteration in Europe. So in frontotemporal dementia, but also in ALS. These are the frequencies in ALS, the familial and sporadic cases. So at that time, we aimed to assess the role and the prevalence of this expansion mutation in a kind of a significant cohort of patients of ALS across Spain. So we managed to join and to analyze about more than 900 ALS patients from 20 centers across Spain, including this city. And what we found is that 7% of ALS patients in Spain have this expansion mutation, and this represents 3% of the sporadic cases and 27% of familial cases. So again, we tried to understand whether the C9orf expansion mutation could modify the clinical course of these patients. And as you see here, the mean age at onset was earlier, a bit earlier, but significant earlier age at onset of patients with the C9orf here in the right. Also, we found that the survival rate was lower in mutation carriers. Again, family history was higher in mutation carriers compared to non-carriers. And finally, and I think this is very interesting again, is that 1/3 of patients with the C9orf mutation also have a comorbid frontotemporal dementia during the clinical course of ALS, whereas only, as expected, 10 to 12% of cases without this C9orf can have frontotemporal dementia co-occurring during the course. So the question was as to whether there is an enrichment of the genetic load underlying this ALS and FTD co-occurrence. That is, so patients with copathology, so patients expressing both disorders, do they have a more genetic load compared to patients without these disorders? And to answer this question, what we did, actually, this is a work from Uriel Dos in our lab. So what he did was we recruited a cohort of 54 patients expressing both ALS and frontotemporal dementia, without the C9orf expansion mutations. These are clinical cases, but also 15 of these cases were pathologically confirmed patients. And what he did was to fully sequence their exome the exome region of these patients, and after a bioinformatic pipeline, we used the variant call file to assess for possible mutations in any of these 30 genes. And intriguingly, and I think this is very interesting, that what we found is that up to 20% of patients co-expressing frontotemporal dementia and ALS have a mutation, a known mutation of any of these genes. TBK1 is the most important genetic cause of patients with ALS and frontotemporal dementia, and we found this huge, I think this is quite important and significant percentage of patients with a mutation in the gene. The second gene in our population is sequestrin-1, which encodes for P62, and this is, and as you see here, we found two patients. Actually, this patient harbored these two mutations. So we found these two patients, the patient number 5 and patient number 6, with the same mutation, the P392L. And as you see here, the prevalence of the rare variant is quite significant compared to the European population. So this chromosome is across our population. So what I'm trying to say is that if you have a patient with FTD and ALS without the C9orf expansion mutation and without the TBK1 mutation, maybe before sequencing sequestrin-1, we should check for this specific mutation because it's quite prevalent in our population. So, and finally, but I think it's also interesting that most of the patients had a presumably sporadic form of disease. So that means that maybe the lack of family history of disease does not exclude the possibility of finding a mutation in these patients. So I'm gonna go to the second section of this talk. So how can we use the molecular genetics for biomarker discovery? And I'm gonna present some very preliminary data from our laboratory on frontotemporal dementia. So as you all know, frontotemporal lobar degeneration term is an umbrella that covers a variety of clinical syndromes with mixed neuropathology and also shared and non-shared genetic factors. So for example, here, patients with the behavioral form of frontotemporal dementia, they can have an underlying neuropathology of tau or in red or TDP-43 in blue. But for example, patients with progressive supranuclear palsy, they typically have tau aggregates in their brain, whereas ALS patients have, as you know, TDP-43. So it's a challenge for now, it's a real challenge to know the underlying pathological substrate of a patient suffering from any kind of frontotemporal dementia. So our aim, there's an urgent, I think there's an urgent need to develop novel biomarkers that can disclose in vivo the underlying pathology in these diseases. So let me go a little bit into our DNA. These are all the annotated loci in our DNA. As you know, we have about roughly 20,000 genes in our genome and 14,000 pseudogenes. But I want to focus your attention on this non-coding region of our DNA, which is the non-coding DNA, and because it represents about 40% of our annotated loci. And these are highly expressed in our brains. So among all these non-expressed, non-coding DNA, so I want now to focus into microRNAs. MicroRNAs are small molecules, RNA, single-strand RNA molecules of about 18 to 24 nucleotides, they have important functions in gene expression. And this is a cartoon trying to to show their biogenesis from the nucleus, of course. They go out of the nucleus to the cytoplasm, and they get embedded in these multivesicular vesicles that fuse with the plasma membrane, and they are expressed to the extracellular space within this round-shaped vesicular of about 50 to 200 nanometers that can go to the outer space and have been proven to be used for as a transfer the information between cells and between organs. So interestingly, many of the genes that have been inequivocally related to frontotemporal dementia or ALS are also related to microRNA biogenesis, thus suggesting that microRNA could have a role in these diseases. So what we did was to try to understand whether the microRNA content within the extracellular vesicles, these exosomes that are found in the CSF of patients with different FTD subtypes, can be potential biomarkers for these diseases. So what we did was to use the CSF from our cohort in the memory unit of São Paulo, and also in collaboration with the Hospital de Clínic in Barcelona. And so we tried to isolate the extravesicular vesicles, and we ran a panel. This is this is a work performed by Laura Cervera. She has a poster here, so if you want more details, you can go to her poster today and tomorrow. So and we ran this panel of 752 microRNAs, and to test for differences between this subgroup of patients. So the first we did was to prove that we were isolating the extracellular vesicles, because this has not been made yet, as far as I know. So in collaboration with Catherine Beyer and Anna Gámez in Germans Trias y Pujol in Barcelona, what we did was to assess for the presence of these tetrasponins. These are the CD63, CD9, and CD81, which we know are very good markers of the exosomes, of the exosomes. And as you see here, we saw as an important signal of these three markers in all the CSFs that we tested. And also we visualized these exosomes through cryo-electronic microscopy, and as you see here in black arrows, we saw this round-shaped vesicles in this patient. So this was strongly suggesting that we were effectively isolating the extracellular vesicles. So we divided the project into phases. The discovery phase, for the discovery phase, we use 10 controls and 10 age-matched frontotemporal dementia cases. And what I'm showing here is that among the 752 microRNAs, we were able to detect about 100 of microRNAs in the CSF, actually in the exosomal fraction of the CSF. So then we went on to the validation phase, and we used 26 candidates, and we analyzed these candidates in a cohort of 30 healthy controls, 20 80 patients, and also we included 72 frontotemporal dementia patients. And among these FTD patients, we divided these patients by the clinical presentation. So in behavioral variant of FTD, in those with PSP or corticobasal syndrome, and those with presumably TDP-43, here in blue, in their brains. So after the analysis, so quality control and calibration and normalization of the signal, so we compared the normalized concentration between these patients and controls. And what we found, I'm going directly to the most interesting data. So we found, for example, here, this microRNA here, you have these normalized concentrations in controls, Alzheimer's disease, behavioral variant of FTD, and also ALS and semantic variant of progressive aphasia. And we found that this microRNA was somewhat increased, its expression was increased in the CSF of these patients with PSP and corticobasal syndrome. But interestingly also is that there were 3 other microRNAs that had a different behavior in their expression, and this was honestly unexpected by us, but we did find this unexpected result that the patients with this Parkinson Plus disease, they express these differences in these microRNAs. And interestingly also is that none of the 34 patients with PSP or corticobasal syndrome expressed this specific microRNA in the CSF. So this is an ongoing project that is being financed by an intramural CyberNet grant, and in collaboration with Maria Bullido and Pepe Rodriguez here. So fortunately, I hope the next year to bring you some mechanistic insight on our findings. So, and last but not least, so as a main conclusion, so I see three main conclusions from my talk. So genetics is of course providing clues about the biological diversity that is present between and within neurodegenerative diseases. So I think that we are starting to understand how genetic factors can shape the clinical course of complex disorders, and I think that new genetic-based tests applied in peripheral tissues can be used as biomarker for neurodegenerative illnesses. This is well proven in fields like cancer, but I think that we should try and push these techniques in diseases from the nervous central system. So last but not least, this is what I wanted to say, that is acknowledgement. I think this work requires a lot of collaborators, and these are our colleagues that have participated in the projects that I have just commented. These are from the ALS field and the movement disorder field. This is also brain biobanks, and especially to Ricard Rojas in our hospital, who is the person in charge of the ALS. And finally, I want to acknowledge, of course, the memory unit in São Paulo, and especially Uriel Dols and Laura Cervera, whose most of the work that I have presented here come from their work. And thank you very much.
Frontotemporal dementia and PSP.
Yeah.
Like frontotemporal dementia and PSP are tauopathies. I don't know if you are looking preferentially to those microRNAs that could affect the expression of tau, like for example 29 or these kind of things. Have you done something like that? Or?
This is something that we have in mind, but we have not checked. So we certainly have to check whether these microRNAs, these candidates that we see, have something to do with tau. That's for sure. I mean, the first thing we want to do is to assess whether the expression of these microRNAs can affect the 4R and 3R isoforms of tau. So this is the first thing we should do, and there are many other projects ahead of us. So we really want to know what's the role of these microRNAs on tau. That's for sure. But we have not assessed yet. Yeah. And we don't have any clue, any previous clue about these microRNAs and tau. To be honest, I don't have any clue. Yeah. Thank you.